Good afternoon, everyone. In this presentation, we are going to learn about the bioindicators in environmental monitoring. Coming to the introduction, the environmental research mainly orient towards the assessment of quality of the environment and its resources. But that assessment plan must have some criteria, and these criteria includes the following, like factors that influence the environment. These factors may be direct or indirect anthropogenic activities such as pollution, land uses changes, or these factors can be natural such as drought, late spring freeze, etc. Second is the criteria that determine the type of intervention which should be employed in this assessment plan. Now, while determining this intervention, determining these plans, interventions such as type of polluting agents or characteristics of environments in which these operations are carried out, or the aim and time required for these assessments are considered. And final criteria is the economic expense, obviously. While carrying out this assessment in conventional system, there are few drawbacks, such as first, like discovery of pollutant in an area does not indicate the possible presence of other pollutants. For example, pollutants such as dioxins, when present in particular area, it does not indicate that there can be other harmful pollutants present in that area also. Or if we consider the opposite fact, that absence of any dioxin compound in any particular area does not guarantee the absence of other particular pollutants. So in this case, we have to go for screening all the type of pollutant present in that area, which is not economically possible. Obviously it's practical, but the type of instruments or the cost will be much more higher, which is the second point. So for this reason, while these environmental assessments are carried out, scholars turn more and more frequently towards the contribution that is made available by scientific sector that is performed in that area. For example, when in a particular area is already assessed for the presence of pollutants such as dioxins, that data is stored and that data is monitored by second batch of scientists to compare the presence or absence, or in other words, the increase or decrease of pollutant in that particular area. So this field is particularly called environmental monitoring, that is the study of quality of environment in which help the assessing the quality of that environmental condition with the help of biological indicators. So these bioindicators or biological indicators provide certain advantages, such as they point out the damages caused in the environment by human activities, and second, they provides the proof of how this environment can be kept under control at lower cost in respect of those foreseen by other methods. Now, each organism within a particular ecosystem has the ability to report the health of its residing environment. So among these specific organisms, also known as those bioindicators, and their association can monitor the change that may indicate the problem within their ecosystem. In this way, these changes can be tracked and economically assessed. Now, coming to the definition, in broader aspect, these bioindicators can be defined as any biological processes or any species or any communities of organism that are used to assess the quality of environment and how it changes over time. But for environmental monitoring purpose specifically, these bioindicators or bioindicating biomonitoring species can be defined as a species or group of species that are used to indicate adverse effect of contamination of pollutants on an environment. Now here is an important point to be noticed, that the term biomonitoring and bioindication, although they are used interchangeably, but they possess some differences among them. Now bioindicators qualitatively assess the biotic responses. That means they qualitatively points out any changes in the environment, such as presence of certain species of lichen like L. conizoides indicates the poor air quality, that is presence of this lichen represent the contamination in the air, while the biomonitoring process quantitatively determine the response, such as reduction in the lichen chlorophyll content or diversity of the lichen species quantitatively determine how this air pollution is affecting that environment. Now here is an example of how this bioindicator species works in an composite environment. Now considering this diagram, it demonstrates the hierarchical level of an ecosystem that respond to anthropogenic disturbances in natural stress. This is an example of a particular aquatic environment where three different species of organisms are considered within a community. This outermost layer, the outermost ring, represent the stress or disturbance causing within that environment. Within that, this colored ring or innermost ring, the color inner ring represent the three individual species of organisms. First one is your cutthroat trout, a particular species, and then second is your salmon fly present within that aquatic environment, and third is a diatom named Phaeodactylum, which these three species of organism reside in a particular environment, aquatic environment. Now the middle ring represents the population of this middle ring represents the population of this organism, and the innermost color ring represents the community in which all these three species coexist. Now these disturbances upon these species can cause the change in their community structure. These disturbances can be anything like disturbance in the energy resources, for example food or light, then biotic interactions such as competition, predation, herbivory, etc., and finally the physical, like the water velocity, substrate upon which the organism attaches, then used for refusal or lay eggs, etc. So when these environmental conditions of or habitat conditions of these species of organisms are disturbed, those disturbances result in increase or decrease of growth or reproduction of these organisms. Now that changes consequently impact the size and productivity of this population and interaction of these species among each other. In other words, when one species falls under this environmental stress, their community structure gets changed, and the change in the number of their individual in a particular population indicates a particular change in the environment. In this way, studying their community structure or their population structure, the quality of this environment can be assessed. Next come the type of bioindicators. Now basically, depending upon their nature of this organism, bioindicators are mainly divided into three main categories: plant indicator, animal indicator, and microbial indicator. Coming to first, that plant indicators are mainly provide very valuable information to predict the status of the environment. Since they are immobile and rapidly obtain the equilibrium with the natural surrounding, The presence and absence of some specific plants or other vegetation in a particular area provides ample information about that environmental health. That means, depending upon the type of pollution or type of stress, the presence or absence of particular species of plant indicate the environmental quality. For example, these lichens generally are found on the trunk of trees. When this quality of the air are changed by the presence of pollutants, different species of lichens appear and some species of lichens disappear upon the concentration of that pollution. That means, for example, if the level of sulfur dioxide or nitrogen increases in a particular region, certain species of lichen will start appearing in the trunk of the trees more compared to some other species. So determining upon that population size of that species, we can determine the type of air quality in that particular environment. Second is your animal bioindicator. Now animal basically indicates the environmental quality depending upon their population structure. Changes in the population may be result of the relationship between populations and food source. If food sources become scarce and cannot provide for the population demand, then the rapid reduction of this population will follow, and decrease or increase population of those animal species can indicate at the quality of that environment. For example, the frogs, which can be used as a bioindicator of quality on environment. Frogs are basically influenced by changes that takes place in their freshwater and terrestrial habitats. So increase in that particular species of frog or decrease in the population can indicate the quality of that environment. Similarly, zooplanktons and aquatic invertebrates also plays an important role indicating the environment habitat condition of their surrounding. Finally, the microbial indicators. Microorganisms are often used in the health indicators of aquatic and terrestrial ecosystem. Due to their rapid rate of growth and due to their abundance, they are easy to test and readily available for environmental assessment. For example, the bioluminescence bacteria in marine environment can be used as a bioindicator. When in presence of toxin, the digestive system of these microbes get hindered, and the amount of light they produce through bioluminescence get decreases. So decrease in the bioluminescence result in the decrease in the bioluminescence can be used as an indication of presence of pollutant in marine environment. Now, depending upon the type of functions, bioindicators can be subdivided into following 4 categories. First one is a pollution bioindicator that detects the presence of any pollutants in the environments. Then environmental bioindicator that detects and or monitor the change in the environment caused by any stressor or pollutions. Ecological bioindicator that detect the change in natural surroundings and their impacts. And finally, the biodiversity indicators that detects the change in biodiversity and the species presence in it due to any natural stress or pollution. Next comes the characteristics of a good bioindicators. First comes the good indicator ability. Any organisms to be a good bioindicator, it must have these 5 characteristics. Under good indicator ability, the bioindicator or the bioorganisms must be able to provide measurable responses. Considering these measurable responses, this organism should not experience any mortality, or they should not accumulate any pollutants directly from their environment. Second is that their response should reflect the whole population, community, or ecosystem response. And finally, the organism should respond in proportion to the degree of contamination or degradation. Second point is the abundant and common. Any organisms to be a good bioindicator, they must have first the adequate local population density. Second, they should be common. Into that area. And third, they should be relatively stable despite any moderate climatic or environmental variability. Third characteristics is the well studied. The organism for to be able to be a good bioindicator, its ecology and life history should be well understood. Taxonomically, it should be well documented and readily available the information to the other scientific community. And thirdly, they should be easy and cheap to survey. Finally, economically or commercially important. The species which are used to be a bioindicator should already be being harvested for other purposes. And lastly, the public interest or awareness of that species should be made clear. Otherwise, due to any anthropogenic activity, the population of these bioindicators may hindered other than pollution stresses. Now, while considering these bioindicators, there are a few techniques used to detect or assess the quality of environment using these organisms. These bioindicators are basically developed specifically to assess the human exposure to the environment, or in other words, the environmental damage caused due to human exposure. Now, calculating this exposure, specific physiological or behavioral changes of this species can be observed by the scientist, but physiological changes where behavioral changes can be observed via statistical analysis or via different methods, but physiological changes should be detected using specific biochemical assays. These bioassays can be carried out in traditional ways or with the application of new biotechnologically derived methods. Under traditional bioassays, bioindicators are introduced into the environment samples, and these researchers observe any chemical changes or any physiological or behavioral changes into that species. For example, measurement of plant root growth is suspected polluted environment, and comparison of the measured growth rate against normal root growth rate can provide information upon how these plant species grow or prosper in a polluted environment. Second, exposure of microorganism to an environment and observation of any changes in the environment related to toxin exposure can such as the presence of stress protein produced when these cells are exposed to harmful environmental condition can give us result on the physiological changes within that microbial organism. Now, although traditional bioassays are easy to use, however, they are time consuming and economically not efficient. On the other hand, to overcome those drawbacks, biotechnologically based bioassays can be assessed. These biotechnologically based method are these biotechnologically based method do not rely on any observation alone, but they set out to create specific reactions that indicate the presence of a specific pollutant or and unwanted microorganisms into the environment. In this way, they are similar to traditional chemical analysis of the environmental sample, but much more efficient in terms of time or economy. Examples of biotechnologically based bioassays are like DNA microarray technology or fluorescent in situ hybridization. The DNA microarray technology, in this method, environmental samples such as water or soil are tested for the actual genetic material of an organism. This form of testing is used to detect any dangerous microorganism into the environment, such as presence of E. coli in water. These microarrays are stump-sized glass or silicon microchips that are embedded with thousands of single-stranded DNA or RNA. In this case, that DNA is taken from that target microorganisms. And second method is the fluorescent in situ hybridization. This is a method for detecting the presence of particular gene in a sample. As a bioindicator, fish can determine whether or not specific microorganisms are polluting in certain areas. This method tests the presence of any microbial gene in an environmental sample. A fluorescent marker is attached to the DNA of the type of microorganism being tested. This marked DNA is now called probe, and this environmental sample containing this probe are fixed onto a slide. And when this probe is exposed to the target samples, if the polluting microorganisms are present into that sample, the DNA will bind to the fluorescent probe, causing the slide to glow in the ultraviolet light. In this way, where a traditional assay takes a few minutes or few hours of testing time, these biotechnologically based bioassays can give results within a few minutes. Now let us discuss the application of various bioindicators and how they help in the detecting environmental monitoring system. First, the common example is the lichens. Now, lichens, due to their sensitivity to different factors of environment, these are considered to be the most appropriate biomonitors of air quality for last 30 years. These are considered the most reliable biomonitors according to their specific physiological, morphological or anatomical characteristics, since they react appropriately and detectably towards the environmental pollutions. Changes in the any air quality can be detected by sensitive epiphytic lichen species. Now, pollutants like nickel, lead, copper, chromium, cadmium, etc. are the results of cement industry. Wind and rain cause the spread of these pollutants to distant area, and these chemicals are usually absorbed by lichens, which can be later detected by these bioassay techniques. Here this table shows the sulfur dioxide air quality and lichen zones. Depending upon the amount of sulfur dioxide present in the air, these lichen zones or this zone are divided into mainly 10 different zones. Each zone corresponds to different sulfur dioxide deposition or different sulfur dioxide concentration in the air. Depending upon the concentration of sulfur dioxide, few species of lichens appear on the trunk, and when the concentration of this sulfur dioxide is very high, few species of lichen completely disappear. Now, zone 1 indicates the heavy pollution of sulfur dioxide. Here no lichen species can survive. Here only algae such as Desmococcus viridis can only grow. On the other hand, the zone where there is no sulfur dioxide, or the sulfur dioxide is under 30 microgram per meter cube, species of lichens tabulated here appear on the trunk. So determining the specific distribution of this species within a particular area, we can determine the quality of air within that particular area. Second is the application of bryophytes while in the biomonitoring process. Bryophytes or moss lack the vascular system and obtain most of their nutrients from the precipitation and from dry deposition of airborne particles. Therefore, tissue contractions are minimally confounded by uptake of mineral elements from soil and subsequent translocation. Here, given an example of a moss named Hylocomium splendens, which was used by a group of scientists led by Hasselbeck to determine the environmental quality in an quality in an area contaminated with heavy metals. Here, the mineral ore is mined from Red Dog Mine, the world's largest producer of zinc, and is trucked along a solitary road to storage facilities on the particular area. So while traveling, translocating these zinc ores, this vehicle travel along a particular road, and this Hasselbeck and colleague examined whether this overland transport was affecting the surrounding terrestrial bryophyte population. Heavy metal content within the moss tissue was compared and varying distance from the road. So as you can see in this graph, metal concentration in moss tissues were greatest adjacent to the road and decreased with the distance. These 4 dots represent 4 different contamination, and as the graph shows, the pollution concentration of this heavy metal decreases with the distance, and thus it validates the hypothesis of those researchers that overland transport was indeed altering the surrounding environment. Here, this concentration of heavy metals was assessed biochemically, and since this method uses quantitative measurement of this environmental monitoring, so here this process can also be referred as biomonitoring using bryophytes as a bioindicator. Finally, the aquatic microinvertebrates can also be used as bioindicator to assess the health of an aquatic environment. Aquatic microinvertebrates possess many of the hallmark traits of good bioindicator. The most common application of microinvertebrates as bioindicator is their community scale. An unimpaired stream or river commonly contain more than 40 unidentifiable taxa, representing a range of habitat preferences and life history strategies. This diversity can capture the responses to different stressors and disturbances, including anthropogenic disturbances. There are several other disturbances are also there, like fine sedimentation, metals, nutrients, and hydrologic alteration. So accordingly, microinvertebrates communities have been frequently used as an environmental, ecological, and biodiversity indicators. Here, the scientist Miller et al. Quantified the aquatic microinvertebrates to identify the threshold separating irrigation water withdrawals. Water withdrawals, when exceeded 85% of the ambient levels, combined with elevated water temperature, reduce the then reduction in food nutrients. It reduces the disturbance intolerant taxa from that area. On the other hand, simultaneously increasing the disturbance tolerant species within that area. Here this line represents the water withdrawal capacity. The green bars represent the abundance of disturbance intolerant taxa. That means when these taxa are this intolerant taxa comes under disturbances, their population decreases, and this blue bar represents the disturbance tolerant or disturbance adapted taxa. Now this study was conducted to see that how much water can be withdrawn for irrigation purposes without disturbing the overall community population of that particular area. Now this study reveals the results of this study reveals that how resource management can be done with integrated response of the entire microinvertebrate community to relate how much water can be taken for irrigation before negative biological responses are seen, while also using the responses of individual taxa to indicate the mechanism of environmental degradation. That means the assessment of this both disturbance intolerant and disturbance adapted taxa can indicate that how much stress here in case of water withdrawal can be provided to the environment so that the ratio of this taxa remain unaltered. So here is the final list of few other bioindicators that can provide valuable information to different level of environmental quality. So bat, indigenous birds and fishes, then freshwater mussels, honeybee, earthworm, phytoplanktons, etc. can indicate the various condition of environmental quality, and detecting or assessing this quality, appropriate measurement can be taken to maintain this proper environmental quality. So finally, let us come to the final part, that's the benefits and disadvantages of bioindicators. Now bioindicators obviously possesses these benefits, like this can be employed at a range of scales from cellular to the ecosystem level to evaluate the health of a particular ecosystem. Secondly, they bring together the information from the biological, physical and chemical components of our world that manifest themselves as changes in individual fitness, population density, community composition and ecosystem processes. And finally, from management perspective, bioindicators inform our action so to what is and is not biologically sustainable. Without these bioindicators, such as the moss in the tundra, the cutthroat in the mountain stream and the canary in the coal mine, we may not recognize the impact of our disturbances before it is too late to do anything to prevent them. However, like most of the system, bioindicators also suffer some disadvantages, like we rely upon the sensitivity of some bioindicators to function as early warning signals. But in some instances, we cannot discriminate natural variability from changes due to human impact, thus limiting the applicability of bioindicators in heterogeneous environment. So we cannot distinguish if the hindrance is due to human activity or it was from natural activity. Second, population of indicator species may be influenced by factors other than the disturbance or stress. For example, like disease, parasitism, competition, predation, etc. And this complicates our picture of causal mechanism of change. And finally, the indicator ability of bioindicator is scale dependent. For example, a large vertebrate indicator such as fish may fail to indicate the biodiversity of local insect community. So this overall objective of bioindicator to assess the quality of environment is useful process, but this can be represent a gross oversimplification of a complex process within that particular community. So this finishes this presentation and this application of bioindicators in environmental monitoring.